Hello everyone, I'm Chaine Wyon, and today we're going to be talking about CRISPR-Cas9, a breakthrough in the discovery of bioengineering. So what is CRISPR? CRISPR stands for Clustered Regularly Interspaced Palindromic Repeats. In short, CRISPR is a gene editing tool for treating disease. Genome editing has actually become a standard of practice in multiple genetic laboratories fixated on mammals. But what does that mean? Essentially, CRISPR has enabled scientists to add, remove, and or even modify a living organism's genetic material. So, let's break down what each letter in CRISPR means. CRISPR can be broken down into two parts, and I feel like it's easier to understand CRISPR by reading it backwards, where we see R, which stands for repeats, or repeated segments of DNA, followed by P, palindromic, which is when you have a sequence of letters that, when read, you can read from left to right or right to left, and so on and so forth. You can see there on the slide, going down from the top left, GCACC is the same as if you were going the bottom right going up, GCACC. This DNA sequence is palindromic. Next is the S, which refers to the short DNA segments that are being repeated over and over and over again. Essentially, what we have here, DNA segments that are getting repeated over and over and over again. But now you see this pattern is I, interspaced, by separate DNA called spacer DNA, which are not the same or identical. And each of these unique spacer DNAs are regularly, the letter R, regularly interspaced in between the repeated DNA. And lastly, these are all C, clustered, as in everything is clustered all together into what is called a CRISPR array. So what is Cas9 then? Well, Cas9 is a bacterial RNA-guided endonucleosis. I know it's a mouthful, but essentially it's a protein. Where an endonucleosis is just an enzyme that specializes in separating nucleotides. And what this means is that Cas9 protein kind of grabs a spacer RNA segment or a copy of the CRISPR DNA and holds it in place with a tracer RNA with the help of a complementary pairing. Its main function, though, is to go through and search for DNA that has its same kind of like scaffolding, what we call, we'll go into a little bit later, guide, the guide RNA. It looks for that, or another thing is PAM, which we'll go into a little bit later as well. It looks for those type of segments in DNA, and it tries to cleave them right where it's at. So how does CRISPR-Cas9 snip and replace DNA? Okay, so now you see when a foreign DNA enters a cell, the Cas9 enzyme will target that foreign DNA if it's able to identify a protospacer adjacent motif region, or a PAM region. A PAM region. If the enzyme recognizes, this Cas9 enzyme recognizes a PAM region in this foreign DNA, it will open up that foreign DNA by using a guide RNA, which we were talking about a little bit before. It's essentially a strand of transcribed spacer RNA, repeat RNA, and a tracer RNA, all in one, made as a guide RNA or a single guided RNA. You don't have to get too far into that, but once the Cas9 opens the DNA, it will check and see if the DNA is complementary to its guide RNA. If it is, the Cas9 will make two cuts above that PAM region and essentially cleave the DNA altogether, causing a double-stranded break. Now, broken DNA will naturally try to repair itself on its own, but this can often lead to mutations or the gene deactivating altogether. But how can we use this system on the cell's DNA and not just foreign DNA? Well, scientists discovered that the reason why Cas9 does not target the cell's DNA is because the Cas9 enzyme is programmed to target the PAM regions. Remember those PAM regions. And essentially, there's no PAM regions in the CRISPR array or the CRISPR DNA, right? So scientists realize that if we can find out what the sequence of DNA is that we want to cut, all we have to do is program that Cas9 guide RNA, that guide RNA in that Cas9, we program it to have the specific sequence that we want gone from essentially like the CRISPR DNA. And then that Cas9 will then go out, hunt down those genes, and we'll cut the genes out of the DNA, which again we'll get in a little further. Additionally, if we want to add DNA, right? So we've talked about taking out genes from our DNA. Now, how do we add genes? Okay, so if we then want to add DNA or a DNA strand into a cell once we've cut something out, we have to essentially, in layman terms, we put this DNA that we have created into the cell, and then it'll go and attach in to where we just recently cut out a DNA. Now, it's a very kind of complex system. We won't go too far into it, but that's kind of how it works. Okay. So now go to the next slide here. Benefits and drawbacks of gene editing. So there's a lot, and I mean a lot of benefits to be had from this discovery. For example, a study done by many scientists in 2020 researched the use of Cas9 and this thing called adeno-associated virus 6 to correct this big, well-known F508 mutation found in the upper airway basal stem cells and patients with cystic fibrosis. Essentially, cystic fibrosis has this F508 mutation going on, and that's what causes a lot of these patients to suffer the way they do with cystic fibrosis. And then once the scientists found out how to kind of target this, they went in and they used this Cas system, and it resulted in a correction of over 30% of the F508 mutation throughout the body, restoring function for the patient and thus supporting gene editing to be a viable treatment for the disease. Further studies are being made every day to find new uses and benefits for this gene editing, and etc. Now, of course, there are always going to be drawbacks to understudy topics. The most blatant drawback of all possible drawbacks is the sheer fact that there just isn't enough research into all the potential negative causes and outcomes of gene editing at this time. And of course, on top of all of that is the essential ethical repercussions that may come as well, which we'll also talk about a little later. So, risk versus reward. For now, let's discuss whether or not the potential risk of editing DNA is worth the potential rewards. So, throughout my research, I have assembled an opinion. And keep in mind, it's only an opinion. My opinion is that I believe that the risk involved with editing a part of your DNA is worth it, as long as the reward is in the realm of possibly treating a life-ending disease or condition such as cancer, cystic fibrosis, AIDS, etc., right? But when it comes to editing your DNA for the sake of cosmetic reasons or for treatments of inconvenient diseases or conditions that are not life-threatening, I believe that at this time, the potential for unknown risks are not worth the potential for unknown consequences. And you know, we live in a world now where we can edit genes, we can edit appearance, we can change eye color, and all these kind of crazy things. So that leads into our next slide here. Do I believe it's ethical? Well, this is kind of a loaded question, right? And this is a very real and current debate in the world right now. The human race as a whole has reached a point in history where the possibility to edit and change our own DNA, the very instruction that makes us human, is right here at our fingertips. So, do I think this is ethical? Honestly, yes. And no, right? Yes, I do honestly believe the future of gene editing will actually advance and propel the human race forward into a world where we may live longer, healthier lives, into a world without worrying about things like cancer, among other diseases. But, on the other hand, what will this also mean for the future of the human itself? Would we stop there? Could we be okay with just conceding after discovering the cure-all to all diseases? What would come next? What sets my stance apart from others is, I believe, my ability to admit my own selfishness. Deep within me, I want to say no, that editing DNA is unethical, only because I know that I may never see the benefits that will arise from it. I was born with the genes I have, and I've had to learn to live with them. They are essentially what made me who I am. As much as I love to think about the idea of the future being full of perfect humans, I just find it hard to stand by the idea of a perfect human, because then that puts me in a world where I'm inferior, I'm below, I'm not perfect, and everyone else is, and that's kind of where the debate is currently right now. It's a pretty heated debate, but that's my little bit on it, and I want to say thank you all for listening in. It's a very short little talk here. There's a couple references I highly recommend looking through. These fantastic articles here, and that's it. I hope everyone has a great day. Thank you.